Hi, good morning. My name is Dr. Ana Soriano, and I am presenting today this project describing the crosstalk between the gut microbiome and intestinal barrier during iron deficiency anemia. I hope you enjoy it and find it useful. First of all, I'd like to start saying that anemia is a public health concern that affects a quarter of the total population, and especially it affects underdeveloped countries, as you can see with the blue colors, dark blue colors in this map. Iron deficiency is the top leading cause of all anemia cases and is also the most common micronutrient deficiency worldwide. At-risk populations of iron deficiency anemia are, because of their physiological circumstances, adult and pregnant women, growing children, and the elderly. Actually, iron deficiency anemia has been associated with detrimental effects in these populations, such as a decrease in skull productivity, increased perinatal mortality, and impaired cognitive function in the elderly. Iron is a very versatile micronutrient, and it can act either as an electron acceptor or an electron donor. This makes it suitable to be involved in a wide variety of biological functions, such as oxygen transport, DNA replications, or synthesis of neurotransmitters. Iron metabolism is a tightly regulated process because both iron excess and iron deficiency is detrimental to human health. Very briefly, the intestinal absorption takes place in the small intestine through the DMT1 transporter. This is a process that is very limited in efficiency because only 10% of the total iron that is contained in the diet is absorbed and uptaken by the enterocytes. Iron deficiency is the most common cause of anemia, and IDA is characterized by an inefficient erythropoiesis, a reduced number of red blood cells, and a decreased hemoglobin levels. The only therapeutical option today to treat IDA are iron supplements that are very limited in efficiency, and they are related to an awful lot of side effects. So oral iron is only absorbed in 10%, and the excess can access the colon when it can affect the gut microbiome and the intestinal barrier. Excess of iron can increase the relative abundance of pathogenic bacteria such as E. coli, and it can reduce the relative abundance of beneficial bacteria such as lactobacillus. In the intestinal epithelium is related, excess of iron can trigger the generation of reactive oxygen species, oxidative stress, apoptosis, and it can damage the intestinal epithelium and the intestinal barrier. So we do know that the gut microbiome and intestinal barrier are affected as a consequence of iron supplements, but we don't know if these are affected during IDA. Studying these aspects can help us reduce the negative impact of IDA therapy on gut health. So just a brief reminder of what the intestinal barrier is composed of. Its function is to prevent the leakage of antigens from the lumen into the bloodstream, and it's composed of three main components: the physical barrier, the biochemical barrier, and the immunological barrier. The first one is also the first one to act, and it has key players such as mucus, intestinal epithelial cells that are sealed by tight junctions, and the microbiome. A healthy microbiome contributes to a healthy intestinal barrier. When the function of the physical barrier is compromised, then the other two components, the biochemical and the immunological one, can act and prevent leaking. When the function of the intestinal barrier is compromised as a whole, there's an increased leakage of antigens from the lumen into the bloodstream, and this can cause systemic inflammation. This inflammation has been associated with a higher risk of miscarriage in pregnant women that are susceptible to suffer IDA, and also it's been associated with higher incidence of autoimmune diseases in children that are also an at-risk population for IDA. So our hypothesis is that the intestinal barrier is impaired during iron deficiency because iron regulates the cell cycle, and therefore iron deficiency can impair the turnover of enterocytes. Moreover, intestinal dysbiosis, which is very likely to occur during iron deficiency anemia, can also alter the intestinal barrier. The gut microbiome plays many roles in the maintenance of gut health, and one of its key functions is microbial synthesis, which consists of providing the host with molecules with limited availability in the environment. One of these molecules are short-chain fatty acids that are synthesized by microbes from fibers. These are mainly acetate, propionate, and butyrate, and they're very well known for their a positive role on the intestinal epithelium. They have immunomodulatory functions, they promote the turnover of enterocytes, and they are involved in the maintenance of the intestinal barrier. With regards to what's been done so far in relation to the gut microbiome and neurometabolism, there's two approaches to address this topic. One is patient-based studies, and these studies have been mainly performed in at-risk populations of IDA, such as children or women. These studies try to investigate the role of the gut microbiome in the generation of IDA. Could the gut microbiome impair iron absorption and be responsible for IDA? The second approach is studies in animal models where iron deficiency anemia is experimentally induced. And these studies try to investigate the response of the gut microbiome to a nutritional or experimentally induced IDA. So is this second approach what we'll be focusing on during this study? And the conclusions of the articles so far are as follows. There's a dysbiosis in the large intestine of these animals, and this dysbiosis is not completely restored after iron repletion. The main limitations is that there hasn't been an analysis of the functional implication of this dysbiosis. So two questions arise in this context, and we will try to respond them during the development of this project. Does dysbiosis occur along the gastrointestinal tract during iron deficiency anemia or just in the large intestine? Does this dysbiosis serve a biological purpose? So the objectives of this study are as follows. The general objective is to understand the interplay between the gut microbiome and the intestinal barrier during the development of IDA, and the specific objectives are: first, the structural and functional characterization of the gut microbiome along the gastrointestinal tract during the development of IDA and the identification of key microbial metabolites, and second, the study of the integrity of the intestinal barrier during the development of iron deficiency anemia and the extent of microbial translocation. Iron deficiency was experimentally induced over a period of 40 days, making use of a diet with normal iron content for the control group or a diet with low iron content for the anemic group. Blood samples were obtained at day 20 and at day 40 to confirm the induction of iron deficiency. At day 40, animals were sacrificed and the following samples were obtained: serum samples and mucus layer samples from all intestinal segments to study the intestinal barrier, and intestinal content samples from all intestinal segments to study the gut microbiome. First, we wanted to confirm that iron deficiency anemia had been correctly induced, and for that we performed a methodological analysis on the blood samples obtained at day 20 and at day 40 from both experimental groups. So we observed that the number of red blood cells, hemoglobin levels, hematocrit, mean corpuscular volume, mean corpuscular hemoglobin, and mean corpuscular hemoglobin concentration was decreased at day 20 and at day 40 in the anemic group. Therefore, iron deficiency anemia was correctly induced. So to pursue objective 1, we first performed 16S RNA sequencing on intestinal content samples belonging to all intestinal regions, and we plotted relative abundances of the 50 most abundant genera in a heat map, where red colors indicate higher abundances. We represent all intestinal region and both experimental groups, anemic represented with an A and control represented with a C. So we can observe that there was no differences in relative abundances in most of these genera in the regions belonging to the small intestine, while there was differences in most of these genera between the anemic and the control group in the regions belonging to the large intestine. Actually, if we focus on the highlighted genera in green, we can observe that there's a more drastic difference between the anemic and the control group in the colon compared to the cecum. So these results suggest that the colon is the region showing the greatest differences between the anemic and the control groups, and therefore the greatest dysbiosis. Next, we perform a predictive functional analysis with 16 sRNA sequencing data, and we performed left-C analysis to identify enriched pathways in both experimental groups. We didn't obtain any enriched pathways in the small intestine, while we did obtain 15 enriched pathways in the large intestine of anemic animals. These KEGH microbial pathways are outlined in this table, and they are ranked according to LDA values. Top ranked microbial pathways are the most enriched ones in the large intestine of anemic animals. And actually, the top 5 are related to short-chain fatty acid metabolism. Next, and in light of recent results, we determine short-chain fatty acid concentration in the intestinal content samples belonging to all intestinal segments. In the case of butyrate, propionate, and acetate, we observe the same pattern where there was a progressive increase of all 3 fatty acids during anemia from the most proximal to the most distal regions of the gastrointestinal tract. And actually, the colon showed the greatest difference and the highest values of all short-chain fatty acids. Therefore, this is in agreement with what we had observed with the predictive functional analysis. To identify potential short-chain fatty acid producers, we performed a multivariant regression analysis to identify the variables contributing or mostly influencing variability of short-chain fatty acids. So for butyric and propionic acid, this analysis showed that variables Clostridium sensu stricto 1 and Clostridium sensu stricto 4 explained around 84% of variability of butyric acid in the colon of anemic animals, while around 55% of variability of propionic acid was explained by Clostridium sensu stricto 1. So these two genera were very likely to be short-chain fatty acid producers. To delve further into the taxonomic analysis at the species level and to also validate the predictive function analysis on 16 sRNA sequencing data, we performed shotgun sequencing on the region showing the greatest dysbiosis, which was the colon. So the functional analysis showed that KEGH orthologs showing differences in relative abundances between the two experimental groups mapped mainly to pathways related to pyruvate metabolism, butyrate metabolism, and propionate metabolism. So these pathways were then colored according to log2 fold change values of differentially abundant KEGH orthologs genes. Squares in this plot represent KEGH orthologs, and the color scale represent higher abundances during IDA with red colors and lower abundances during IDA with blue colors. We can observe that there's an increase in KEGH orthologs related to the synthesis of succinate and acetate, as outlined with the blue and red and green arrows. Analysis of the butyrate metabolism pathway revealed an increased abundance of genes related to the synthesis of butyrate, as outlined by the blue, orange, and green lines. And lastly, analysis of the propionate metabolism pathway also revealed an increased abundance of genes related to the synthesis of propionate, outlined by the green, orange, and blue lines. Taxonomic analysis at the species level revealed that Clostridium species were enriched in the colon of anemic animals. Pearson correlations were calculated between these statistically significant species and short chain fatty acid levels, and they were plotted in a heat map. In this heat map, brown colors indicate negative correlations and purple colors indicate positive correlations, and we can see that Clostridium species are positively correlated to propionic and butyric acid. Therefore, results obtained with predictive functional analysis and 16S RNA data are validated using shotgun sequencing. To pursue objective 2, which was the study of the integrity of the intestinal barrier and the study of the microbial translocation, we performed mRNA sequencing on the colonic mucus layer. As a reminder, the colon was the region showing the greatest dysbiosis, and the gut microbiome is a key component of the intestinal barrier. So the region with the greatest dysbiosis was very likely to also have alterations in the intestinal barrier. That's why we chose the colon. We performed a GCA or gene set enrichment analysis on mRNA sequencing data using gene ontology as a database, including its three categories: biological process, cellular components, and molecular function. We plotted the results using the Revigo tool, which showed only the statistically significant parental GO term differing between the two experimental groups. In this bubble plot, each bubble represents a deregulated GO term. In this slide, we can see the upregulated ones, and we can observe that there's GO terms related to lipid metabolism, and they are highlighted in blue. There's also GO terms that are related to host microbiome interactions, which are highlighted in red, and there's also a bunch of terms that are related to the mitochondrial electron transport change, and these are highlighted in yellow. Among the downregulated processes, there's GO terms related to the synaptic signaling within the enteric nervous system, which are highlighted in green. There's also terms related to the development of the digestive tract, which are highlighted in black. There's also terms related to cell-cell junctions and the organization of the cytoskeleton, which are highlighted in orange. And the ones that are highlighted in purple are related to the organization of the extracellular matrix. So the downregulation of these GO terms indicate an underdeveloped epithelium with underdeveloped cell-cell junctions, and this indicate an impairment in the intestinal barrier. So lastly, to study microbial translocation, we used serum samples and we determined two biomarkers, LPS and bacteria-specific immunoglobulins. In the case of LPS, we obtained higher values in the serum belonging to the anemic group, confirming an increased microbial translocation. In the case of bacteria-specific immunoglobulins, we used two experimental designs. In the first case, what we call per feces, each serum belonging to each experimental group were tested against bacteria isolated from the feces belonging to the same experimental group. So serum belonging to the anemic group were tested against bacteria isolated from the feces of the anemic group, and the same for the control group. So in this case, we observed an increased immune response in the anemic group. However, this increased autologous immune response wasn't reproduced when we used the second experimental design, in which sera belonging to both experimental groups were tested against bacteria isolated from feces belonging to the control groups. This indicates that the increased autologous immune response that we observed in the first case is being produced against bacteria that are only present in the feces of anemic animals and not the feces of the control animals. And these bacteria are very likely to be translocated. All in all, results from microbial translocation supports a dysfunctional gut barrier during IDA. To discuss briefly results obtained, just mention that iron is a micronutrient involved in the regulation of cell cycle and DNA replication, and this justifies the underdeveloped epithelium that we observed in RNA sequencing data. Iron is also involved in neurotransmitter synthesis, which is in agreement with the decrease in uptake signaling that we observed. And iron is also essential for collagen metabolism and therefore is involved in the organization of the extracellular matrix, which justifies that there's a decrease in extracellular matrix associated genes and proteins during IDA. The increase in short-chain fatty acid that we observed could be a trade-off mechanisms to make up for these altered gut barrier, since they are known to promote turnover, promote neural survival, neurogenesis, and also they maintain HIF-1 alpha dependent functions in the intestinal barrier. Lastly, a huge thank you to my research group, my department, and all the entities that provided funding for this project.